Hello students, welcome back to the channel. In this video, we are going to study about active transport. Active transport is a method in which substances are moving against their concentration gradient. Because they are moving from low concentration to high concentration, we have to use energy. ATP is used. That is why we say it is active transport because energy is used. And the protein which is involved, two types of proteins are involved. One is carrier protein, another is pump protein. Means you can say carrier proteins with a usage of ATP, we call it as pump protein only. So to pump a solute against its concentration gradient, cell has to spend energy and we say it is an active transport. Now the transport proteins which are involved in this, they are carrier proteins. They are carrier proteins and they are not channel proteins, because channel proteins, they are allowing the solute to go down their concentration gradient, but carrier proteins are picking them and transporting them against their gradient. Active transport also allow the cell to maintain internal concentration of solute different from the surrounding. For example, when we see a cell, we find that inside the cell, potassium concentration is high and sodium concentration is low. So as compared to its surrounding, animal cell will have high concentration of potassium and low concentration of sodium. How they are able to achieve this? So plasma membrane and especially the carrier proteins, that pump proteins, they are helping to maintain these steep gradients by pumping sodium out and potassium into the cell. And for this, ATP is used. Now, let me explain you with the example. See here. See, this is one transport protein. Okay. And outside, sodium is extracellular fluid. Sodium is high here and potassium is low here. Inside, potassium is high and sodium is low. This is the one basic you should remember. Now, how do they maintain it? We discussed now, they are when plasma membrane protein are helping it. Now, see here, 3 sodium ions are binding. So, a specific protein is there where the 3 sodium ions are binding on the inside. Now, binding of sodium ions will cause phosphorylation of ATP. Okay, they will actually, after binding, ATP will go for phosphorylation. Phosphorylation means ATP will change into ADP plus inorganic phosphate. Is it clear? The first binding will cause phosphorylation. Then what will happen? See, it will cause the protein to change its shape. Phosphorylation will cause the protein to change its shape, expelling 3 sodium out. Is it clear? Expelling 3 sodium out. Now, in outside, potassium is low. So we have to bring the potassium inside now. This is the way how they are maintaining it. So what happened? 2 extracellular potassium ions will bind to the region now. And this caused the release of the phosphate group. After binding, the phosphate group is now released. Phosphate group will release. See here. Binding is causing the phosphate group to release. Is it clear, beta? Now, after this, what happened? Loss of phosphate group means it will come back to its original shape. And what's the original shape? Means it will be opening towards the intracellular side, causing the release of the potassium ion into the intracellular space and is ready to bind next 3 sodium ions. So this is the way how active transport is done. They are moving the ions against the concentration gradient with the help of ATP. And the protein which is taking part, that is a carrier protein, but because it is powered by energy, means powered by ATP, we say these are pump proteins. Is it clear? These are pump proteins. So I think so sodium potassium exchange pump topic might also be easy for you. Now this is a quick review for entire flow chart. You can see here. Check beta, cytoplasmic sodium will bind. Affinity for sodium is high when the protein is in this shape. Then sodium binding will cause the phosphorylation of ATP. Phosphorylation will lead to the change in the shape of a protein, reducing its affinity for sodium, which is released outside. New shape is having affinity for potassium, and potassium binding of potassium will trigger the release of phosphate ions. Phosphate groups are released and it is coming back to the normal position, original shape, which is having low affinity for potassium. So now potassium is released and again its affinity is high for sodium. So the cycle continues. So the two shapes, they have different affinities for sodium and potassium, and ATP is the one which is changing the shape by transferring phosphate group or by removing the phosphate group. I hope so. Up till here, this topic might be easy for you. So in this video, active transport topic is discussed. I hope this video is helpful. Thanks for watching.